My name is Reiko Kuroda. I'm a professor at the Research Institute for Science and Technology. I work in the field of chirality, which refers to left and right-handedness. Chirality is ubiquitous in nature. It exists at the microscopic level and the macroscopic level. It exists in both the biological and non-biological domains. Chirality is an endlessly interesting topic. I coined the term chiromorphology from the words chirality and morphology. We use it to describe the concept of chirality of shape across the domains, with a view to linking microscopic and macroscopic phenomena. Chirality is particularly important in life, as the biological world is, at the molecular level, homochiral, meaning that all living organisms on Earth use exclusively the right-handed deoxyribose for DNA and only the left-handed amino acids for proteins. My research covers primarily two topics. One is molecular chiromorphology research, to investigate solid-state chiral chemistry, because molecular interactions and hence chiral recognition are much stronger in a solid-state environment than in solution. Quartz crystals are chiral, but table salt crystals are non-chiral in shape, and the crystal chirality stems from the chirality at the molecular level. We have been studying science behind the link between molecules and crystals through chirality, and applying it to novel solid-state chemistry. We have discovered several exciting phenomena, such as facile chiral resolution of secondary alkyl alcohols, notorious for their difficulty in left-right separation, simply by way of making supramolecular crystals, chirality recognition, generation, and amplification during crystallization, and molecular rearrangement in the solid state. Chirality recognition and amplification are key to understanding how a homochiral biological world was established 4 billion years ago from a world that contained almost equal numbers of left and right-handed molecules. This basic scientific research also has important implications for everyday life. As life is homochiral at the molecular level, left-handed and right-handed molecules often have entirely different end results, such as in terms of drug effects, taste, or fragrance, similar to how a right foot fits well in the right shoe, but not in a left shoe. One famous example is thalidomide. Developed as a sedative and said to help with anxiety, stress, and nausea, thalidomide taken during pregnancy led to thousands of children with birth defects. The left-handed nature of the thalidomide molecule is thought to be responsible for this disaster. To study chirality in the solid state, we must have information on chirality, for example, to elucidate structures of compounds in that phase. However, as solid-state chirality cannot be measured by commercially available instruments due to macroscopic anisotropies that are intrinsic to the solid state, we developed unique spectrophotometers. With these, we have obtained new knowledge, including about aggregation of beta amyloid proteins relevant to Alzheimer's disease. My other core research topic is organismal chiromorphology, to link genes and proteins and body-handedness of living organisms. Snail coiling is determined by a single gene locus. My research group has revealed the link among the handedness determining gene, cytoskeletal dynamics in a very early embryonic stage, and the snail shell coiling. Further, by mechanically altering embryonic chirality at this particular embryonic stage, we were able to create healthy creatures that are the mirror image of the original. This was achieved by moving the nodal gene expression site to the mirror image site within the embryo. Moreover, their offspring return to normal, indicating that chirality manipulation affects gene expression but does not change the gene themselves. Also, the homologs for this gene is known to be important for left and right-handedness formation in vertebrates. For more than a century, researchers have sought the gene that determines snare-handedness. We hope to identify the gene soon, and that our research will help to elucidate common mechanisms for body-handedness determination in animals, including human beings. So you can see why chirality is both important and interesting. And we plan to pursue this important and interesting chiromorphology research both in chemistry and biology, as we strive to make even more discoveries.